chapter. So, but all of chapter 13 will be on the test. So the brain is made up tissue-wise. We've already talked about the cells that make up nervous tissue. But if you do a cross section of the brain or a dissection of the brain, you will notice that there are differences as far as the colors are concerned of the tissue that you are seeing. So we call that different tissue gray matter and white matter. The reason white matter is white is because of the presence of myelinated axons. That's what gives it a white appearance. And then gray matter is primarily made up of dendrites and cell bodies, and then some portions of axons that are not as heavily myelinated. When we look at the brain, you'll notice that there is gray matter on the outside and white matter on the inside. And then the opposite holds true for the spinal cord. If you do a cross section through the spinal cord, the white matter you find on the outside and the gray you find on the inside. When it comes to the ways in which we structurally discuss the brain, the upper portion of, or I should say the superior portion of the brain, the portion of the brain that has the very visual convolutions that we're so used to seeing, we call that the cerebrum. The cerebrum itself has two hemispheres. We'll be going over structures that are found there in good detail coming up here. And then we also functionally organize the cerebrum into 5 different lobes. The second major region, so the first major region of the brain, again, is the cerebrum. The second major region of the brain is what we call the diencephalon. So the diencephalon is made up of the thalamus and again, I'll be showing you this in a moment. The thalamus and the hypothalamus and the epithalamus. And then the brainstem is the third region. The brainstem is made up of the midbrain, the pons, and a structure called the medulla oblongata. And the medulla oblongata is the last portion of the brain, the most inferior portion of the brain that's going to lead into the spinal cord. You can see that on this picture pretty well. And then the fourth major region of the brain is what we call the cerebellum. The cerebellum, when you look at the brain, either a model of the brain or images of models, you'll notice that it has kind of a different texture to it. It's not made up of this folded tissue. You don't find those convolutions. The tissue itself almost has kind of like a striated appearance, gross anatomically speaking. The cerebellum plays an important role in when it comes to balance and posture and many actions that are not under conscious control. Many of those things are kind of fine-tuned by regions of the cerebellum. And then the last thing I just want to mention, which we will talk about at the end of this chapter, is that when it comes to nerves, right, we're going to talk about spinal nerves in the spinal cord chapter, but within the central nervous system, we have 12 pair of cranial nerves. They're called cranial nerves because they originate in the brain. However, they leave the brain and leave the central nervous system. And so technically they're a part of both the central and the peripheral nervous system. So as far as the functions of those 12 cranial nerves go, I do expect you, and so for both lab and for lecture, you are going to need those to know those 12 nerves, and we number them using Roman numerals, right? So and the acronym CN, which is cranial nerve. So CN Roman numeral 1, CN Roman numeral 2, 3, etc., all the way up until 12. And then there's a variety of different mnemonic devices out there to help you remember it. You know, the one I always use is the on occasion, our trusty truck acts funny, very good vehicle anyhow. Again, we'll be talking about this at the end of the chapter. I'm expounding on it right now because I think this is, when it comes to the cranial nerves, it's just, it's pretty much rote memorization. And so I think it's good to start committing that to memory now, right? Make yourself, learning the cranial nerves is great. Flashcards work great for this. And then just kind of thumb through those flashcards when you're watching, even unwinding, watching TV, thumb through them, because you are expected to know them. You're expected to know the Roman numeral associated with them, and then you're expected to know their function. Um, so like I said, I bring it up now so that you can kind of start in on learning that material. Okay, so this is what the brain would look like. And I recognize that you guys do not have the luxury of doing a wet lab and doing the sheep brain dissection and looking at the models, but the models that you find from Practice Atlas are really good. The images are really good, and the APR images are great too. So this is, if you were looking at the outside of the brain, this is what the brain would look like. And what you would notice right away, as far as the landscape, if you will, of the brain is concerned, is that again, it's convoluted, it's folded. Each one of those ridges created by the folds are what we call a gyri or a gyrus. And then the folds themselves, of course, are going to create grooves. So the shallow, these shallow grooves are what we call sulci, or one is a sulcus. And then there are also some deep grooves, and those deep grooves are called fissures. And then as far as neurulation goes, so this is just a kind of a very quick overview on embryological development. What I want you to know as far as embryological development of the central nervous system is concerned. If you go on and you take a developmental biology, you'll go into a great deal, you'll go into a great detail about these processes, but I just kind of want to run you through the basics. So neurulation itself happens early on, embryologically speaking. Neurulation begins around the 3rd week, which is a time that many women don't even yet know that they're pregnant. And that becomes really important in the sense that during this portion of the development, if a pregnant woman is exposed to what we call teratogens, which are entities that can cause birth defects, if you're exposed to them, if you're exposed to teratogens during this 3rd week, it can have, if, you know, and those teratogens are going to have an effect. What those teratogens will affect likely will be portions of this of the nervous system that are developing. So what happens around this 3rd week is you get the formation of what we call the neural tube. The neural tube is formed from the ectoderm. So if you can remember back to when we were learning about those primitive tissues, the endoderm, mesoderm, and ectoderm, the ectoderm is where you get many of the structures that are part of the nervous system. So the endoderm forms the neural tube, which is what you're seeing here. And again, if this event is either disrupted, or if the pregnant woman is exposed to teratogens during this time, or is lacking in certain there are certain things that are lacking in her diet, such as folic acid, that can contribute toward possibly causing what are called neural tube defects, because it is the neural tube that, as it expands or grows, is going to become both the brain and the spinal cord. So all of the central nervous system tissues derive from this neural tube. Okay, so a little bit on what we call traumatic brain injuries and the differences between contusion and concussion. So traumatic brain injury is something that's gotten quite a bit of press, I would say, in the last 20 years, because we are learning more and more about the effects that, for example, concussions and repeated concussions can have on the brain. So TBIs are included, it's the umbrella term to include any injury to the brain tissue. Concussions are relatively common when it comes to injuries. Concussions are classified as being a temporary loss of consciousness after some kind of insult to the brain or to the head. Oftentimes when people wake up from concussions, they're drowsy and they're confused. And concussions typically tend to resolve themselves pretty quickly. But again, we're learning more and more about this summative or cumulative effect. that concussions, that sustained concussions can have when it comes to long-term ramifications, such as the development of early onset dementia and things of that nature. Um, contusion is a contusion is different from a concussion in that it is you get not that you can't get bleeding with a concussion, but concussions historically speaking have not been associated with bleeding. Contusion is when the head is injured and there's actual bleeding that occurs, so you get kind of like a bruise um, of the brain, but you're still unconscious, um, for a relatively short period of time, no more usually than, um, you know, 8 minutes at the most. And then the second impact syndrome, again, what I want you to know about that is just that it's tied to this idea of if the summative effect of concussion. So second impact syndrome just says if somebody has a brain injury, they're much more likely to have a second, so a reoccurring brain injury in that first year. And oftentimes that second brain injury, even if it's not even if it's the type of injury that would typically not cause tremendous damage for individuals that have already had a TBI, it can result in major brain bleed. Okay, and then for neural tube defects, again, these defects occur as the neural tube is forming, so early on uh, in the pregnancy. And there are many, many, many different things that can cause neural tube defects. And many of those have nothing to do with exposure of the pregnant woman to, or lack of exposure to certain vitamins or exposure to tritogens. However, we do know that there is a increased risk of neural tube defects when folic acid, when there's not a sufficient intake of folic acid. And then some of the types of neural tube defects I want you to be familiar with. So I want you to know what anencephaly is. Anencephaly is a condition in which there is a lack of all of or a significant portion of brain tissue, along with not having the bones that make up the cranium. Anencephaly, again, there's degrees of anencephaly, but unfortunately, it's typically a condition that is not associated with life. And then and by life, I mean with longevity. The individuals with anencephaly don't tend to survive for very long after birth. Um, and then spina bifida. So spina bifida, I want you to know that there are two types and what characterizes spina bifida. So spina bifida occurs when there is failure of closure of the caudal portion of the neural tube. And there are two broad types of spina bifida. So spina bifida, what we call spina bifida cystica, it occurs when it's the more serious of the two types of condition. And it occurs when the neural tube doesn't close, and then the cerebral spinal fluid essentially forms a cyst. So you can see the cyst, or you see an opening somewhere along the vertebral column in the newborn. And this is the kind of spina bifida that oftentimes is associated with paralysis because depending on where the opening is located, all of the spinal nerves that are inferior to that opening are going to be not functioning correctly for the most part. And then spina bifida occulta is interesting in the sense that oops, sorry guys. This is a condition in which there's a defect, particularly a defect of the bony arch and of the vertebra. But individuals with spina bifida occulta oftentimes are completely asymptomatic. And in fact, this is the one indication of occulta is these infants are born and there can be a little tuft of hair that grows right where, when they're born, right where the defect occurs. But if that hair is not present, oftentimes this condition is, people are completely asymptomatic, oftentimes. Okay, so I do expect you to know the secondary brain vesicles and then the structures that these brain vesicles become. So the secondary brain vesicles are formed by the end of the 5th week, and there are 5 of them. So we have what we call the telencephalon. The telencephalon right here becomes the cerebrum. Then we have the diencephalon, and the diencephalon, which is nice, becomes the diencephalon. It becomes the thalamus, the hypothalamus, and the epithalamus. The mesencephalon becomes the midbrain. The metencephalon becomes the pons and the cerebellum. And then the myelencephalon becomes the medulla oblongata. So this is also material that lends itself really well to flashcards. Okay, so another clinical view that is associated with brain formation is a condition called lissencephaly. Lissencephaly is relatively rare, and it's also called smooth brain. So with lissencephaly, what happens is individuals with this condition, their brains are smooth, so they're missing gyri and sulci, and those gyri and sulci, anytime you see a folding or a convolution in tissue, kind of remember, take note that what that does effectively is it increases the surface area. And so having our brains have all these convolutions increases the surface area available for neurons to engage in synaptic activity. So individuals with lissencephaly, their synaptic activity is disrupted and they tend to suffer from terrible seizures, and be cognitively impaired, and just kind of like spina bifida, there are degrees of lissencephaly. So there are some individuals who live quite a long time, I think into their 30s with this condition. Okay, so again, when we talk about the brain and the spinal cord, gray matter for the brain is found on the outside, or it is more superficially located than white matter. For the spinal cord, the white matter is found on the outside and gray in the middle. And as I mentioned on that first slide, gray matter is going to be made up mostly of cell bodies and dendrites and unmyelinated axons. And then the white matter is white because of the presence of myelination. So it's made up of myelinated axons. If you look at a cross section through the brain, what you notice within the white matter are these islands, if you will, of gray matter. And those islands are called cerebral nuclei. And these, just like what we find that makes up gray matter, these islands are essentially clusters of neuron cell bodies that are found within the brain. And they are there's functional significance associated with them that we're going to talk about when we get to that slide. But just kind of take note, because again, we're going over the gross anatomical characteristics or features of the brain. So take note that there are these islands of gray matter called cerebral nuclei deep within the white matter of the brain. Okay, so the meningeal layers. The brain itself is, of course, encased by bony tissue, and we call that bony tissue the cranium. And there are tremendous benefits to having the brain located in having a bony encasement. And there are also limitations and those limitations become specifically apparent when we see swelling in the brain, because if brain tissue swells, there's literally nowhere for it to go, because it's in an enclosed bony cavity. So kind of more on that to come when it comes to various clinical presentations that are associated with brain swelling. But circling back to the meninges. So the meninges are these connective tissue protective sheaths that cover the brain. And there are three of them. The outermost layer is what we call the dura mater. That term means tough mother, and that's because this connective tissue sheath is the thickest, definitely, of the three meningeal layers, and it's relatively tough. If you were to do a brain dissection, if you were to have done the sheep brain dissection, you'd be able to see the dura mater on the sheep brain, and you have to cut it off. And again, it kind of gives you an appreciation for how tough and durable that dura mater is. Deep to the dura mater is what we call the arachnoid mater. The arachnoid mater, and you can see it in this picture here, is called the arachnoid mater because it has these little appendages, if you will, that hang down, and they look like spiders' legs. As we get into the deeper and deeper layers, the layers themselves become thinner. The mater or layers become thinner, and much more delicate. So the arachnoid mater is a relatively delicate layer, but it has these, again, these like little appendages that hang down, and they play an important role when it comes to those, along with, sorry, I should mention, there are also these outcroppings that occur that play an important role in the reabsorption of cerebrospinal fluid. And I'll say this right now, and I'll say it again going forward, but cerebrospinal fluid circulates through the brain in the subarachnoid layer, or in the subarachnoid space, right? So sub meaning beneath. So the layer beneath the arachnoid mater, which is right here, this is the subarachnoid space, and this is where cerebrospinal fluid flows. And then the innermost layer is what we call the pia mater, and the pia mater is essentially invisible to the naked eye. And that's because it's so thin and delicate. It clings to the surface of the brain, and it follows the convolutions. And so you can see in this picture here, this would be the pia mater, again, following the convolutions of the cerebrum. Okay, so that's what this slide addresses. I just said it a little bit early. The other thing I want to bring to your attention, so those, sorry, those little appendages, again, that come off of the arachnoid mater, those, along with the villi, are involved with the circulation and reabsorption of cerebrospinal fluid. There are also spaces between these maters that become clinically very relevant. And in an intact specimen, the spaces are potential spaces, so they're not real spaces, but they are spaces into which blood can accumulate if there is a brain bleed. And in that case, they become actual spaces. So the clinical, and there are, so we have a subdural space, right? That would be the space between the dura mater and the arachnoid mater. The sub tells you it's the space below the dura mater. And then I already made mention of the subarachnoid space, which is below the arachnoid mater, between the arachnoid and the pia mater. And that's the space through which cerebrospinal fluid circulates. We also have another important feature that in the context of what we're talking about that I want you to be familiar with are the dural venous sinuses. So these sinuses, as you can see, here's an example of it. Whoops, my cursor. I have a section through one right here. These are essentially, it's vasculature, and these are the veins that drain the blood from the brain. So the blood that is drained by the dural venous sinuses is going to be deoxygenated blood that's going to get dumped back in venous circulation and make its way back to the heart so that it can go from the heart to the lungs to be oxygenated. And then the last space is the epidural space. So the epidural space tends to be, I suppose, a little bit less clinically relevant in the sense it doesn't tend to be a space in which blood accumulates, although blood can leak into that space and accumulate. But it is another one of those potential spaces that exists. You'll notice that there is no epi or sub pia space, and that's just because that layer, again, is so thin and delicate that fluid doesn't even excuse me, have an opportunity to accumulate in the spaces either above or below it, because at that point the tissue would just be destroyed. So that kind of nicely brings us to this slide, which has to do with the clinical relevance of those various spaces. So here are subdural and epidural hematomas. So subdural are, not that they're common, but they're, if someone sustains a head injury, I guess they're relatively more common. Subdural hematomas can be insidious, and that these are slow brain bleeds. So these are the bleeds in which they tend to be venous bleeds, and blood will accumulate in the subdural spot, in the subdural space, and then these individuals are the ones that you hear about who get into a skiing accident, and then they're fine, and then all of a sudden 10 days later they have a seizure, and die, or something along those lines. And that's again because these are slow brain bleeds. Epidural hematomas tend to be arterial bleeds, and so these are the type of bleeds that they're very fast bleeds. They're oftentimes associated with a crack in the skull. Individuals with epidural hematomas lose consciousness pretty quickly, and these are the people, again, that it's obvious they have sustained an injury, and this type of injury oftentimes requires surgical intervention to remove the blood. Yeah. Okay. And then meningitis is another important clinical view associated with the central nervous system. Meningitis, the itis always means inflammation of. So this is inflammation of the meninges. Can be due to some kind of pathogenic assault, can be viral in nature, bacterial in nature. But at the end of the day, this is problematic, again, because anytime, well, first of all, because of the blood-brain barrier, the central nervous system is an area or it's a system in the body that is not very good at addressing infections. The immune system has a difficult time gaining access to the central nervous system. So that is one reason why it's problematic. And then, of course, the other reason is that anytime you have inflammation, you're going to have swelling. And as I mentioned, central nervous system structures are encased in bone, right? When we talk about the spinal cord runs through the vertebral column, which is made out of bone. The brain is found in the cranium, which is made out of bone. And so when inflammation happens, that tissue is, if there's no surgical intervention, that tissue is it swells and it presses against the bony encasement, and tissue death starts to occur. And or, maybe even worse, within the brain, you can get invagination of brain tissue out of the foramen magnum, which is the large hole through which the spinal cord runs. Again, that would cause tissue death, and oftentimes death of the organism. Okay, so ventricles within the brain. Ventricles are spaces through which cerebral spinal fluid flows. We talked, when we were going over the glial cells, we talked about ependymal cells, which are a type of glial cell for the central nervous system, and how those are cells that line ventricles and are associated with capillaries, and those are the choroid. We call those associations, or that association, of the choroid plexus, and that is the structure that's responsible for making cerebral spinal fluid. So that, in the context of what we were talking about then, I was talking about the lateral ventricle, which is the largest space in the brain, and that is where cerebral spinal fluid primarily is made. And then we have some other ventricles or spaces as well. We have what we call the third ventricle. The third ventricle is found in the diencephalon. And then we have the fourth ventricle, which is found between the pons and the cerebellum. It's difficult to see on these illustrations, I recognize. But I'll show you when we go over cerebral spinal fluid through the brain, where those ventricles are found. Okay, so some characteristics of cerebral spinal fluid. It's a clear, colorless liquid that, as far as composition is concerned, shows a lot of similarities with blood plasma. It circulates throughout the ventricles and, again, throughout the brain in the subarachnoid space. And what this, functionally speaking, what the cerebrospinal fluid does, very importantly, is it allows the brain to kind of float in the cranial cavity. Again, because the brain is encased in bone tissue, if we didn't have cerebrospinal fluid and you hit your head, as we all do, and especially when we're developmentally when we're toddlers, as we know, fall and hit their heads quite often. And so having the brain floating, if you will, in cerebrospinal fluid gives it buoyancy. And so, and if you've ever been in a swimming pool or in the ocean or in a body of water, you know that if you try and move your arm, for example, underneath water, it moves much slower because of all of the drag that's created by the water you're moving through. So the same thing goes for the brain. If you hit your head and your brain moves back and forth, you're going to have less trauma done to that tissue if it's moving back and forth in water, because that's going to help to decrease the impact, the velocity of impact of the brain tissue on the cranium when you hit your head. So that's kind of the buoyancy and protection aspect of the cerebrospinal fluid. And then the cerebrospinal fluid also helps to create environmental stability for the central nervous system. So plays a role in helping to regulate temperature, to provide nutrients to the brain, or I should not just say the brain, to the central nervous system tissue, to remove waste products, to allow for chemical messengers, various types of chemokines and even neurotransmitters in the synaptic cleft. All of those types of processes occur better because that tissue is immersed in cerebrospinal fluid. So it's very, very important when it comes to maintaining homeostasis of the central nervous system. Again, cerebrospinal fluid is formed by the choroid plexus, which is the association of ependymal cells with blood capillaries, and the vast majority of cerebrospinal fluid, as I mentioned, is made in the lateral ventricles. And I apologize, I'm losing my voice. So hopefully everybody can understand me. So when it comes to circulation of cerebrospinal fluid, I expect you to know this. So cerebrospinal fluid, again, is primarily made in the ventricles, and specifically the lateral ventricle. Hold on one moment, you guys. I have to move this out of the way. It then flows from the first ventricle into the third ventricle come on, cursor. Here you go. It flows from the lateral ventricle, where it is primarily made, through there are two channels that are associated with cerebrospinal fluid flow. So the first is what we call the interventricular foramen. The interventricular foramen is called that because it is a ventricle that's connects the ventricles. So cerebrospinal fluid goes from the lateral ventricle to the third ventricle through the interventricular foramen, and then it flows from the third ventricle to the fourth ventricle through what we call the cerebral aqueduct. And then from the fourth ventricle, it flows throughout the subarachnoid space. So, essentially cushioning the brain. And then it's ultimately reabsorbed by these villi, the subarachnoid villi, that then, again, dump cerebrospinal fluid back into the dural venous sinus, or back and otherwise dump it back into venous circulation. Okay, lots of clinical views when it comes to this material. So hydrocephalus is a term that's used the word literally means water on the brain. So due to a variety of different underlying etiologies, we can develop hydrocephalus. So brain injuries can result in hydrocephalus. Infants can be born with hydrocephalus, but hydrocephalus is used to describe an accumulation of cerebrospinal fluid that is disrupting homeostasis. So that makes it pathologic. Again, variety of different underlying reasons why somebody can develop hydrocephalus. but hydrocephalus, if it becomes problematic, is treated usually by implanting a shunt and draining the excess cerebral spinal fluid so that it doesn't increase pressures in the brain to a point that is problematic. Okay, so the blood-brain barrier, we've talked about a little bit already, right, when we were talking about astrocytes. So this is a highly selective barrier that's made up of the astrocytes, and the astrocytes wrap around. They have those perivascular feet that wrap around capillaries, and that's what forms the barrier itself. The barrier, it's the blood-brain barrier, again, it's a highly selective barrier. So there are substances that can get through, and those substances are namely lipid soluble substances, so non-polar, um, and typically relatively small. So drugs, many of the non-polar drugs that are delivered can get across the blood-brain barrier. And then another important feature when it comes to the blood-brain barrier is that it is missing in 3 different locations within the central nervous system. And those places are going to be the pineal gland. So anytime we talk about an endocrine structures are those structures that make hormones, make and secrete hormones. And so you'll notice that 2 of these structures where the blood-brain barrier is missing are endocrine structures. And that's because having a blood-brain barrier around those structures would impede the ability of the hormone to get into circulation and travel, which is what hormones do. So the places we find the blood-brain barrier missing are the pineal gland, which is responsible for secreting melatonin, which is a hormone that regulates circadian rhythms. And then the hypothalamus, which is, we'll talk a lot about the hypothalamus when we get to, and we'll talk about it a bit in this chapter, but a lot when we get to the endocrine system. But the hypothalamus is it's the control center for the endocrine system. It's the control center for the body in many ways. It plays an important role in regulating temperature and thirst and a lot of very vital functions that we'll get to. And then the choroid plexus, which again, that is the structure that makes and secretes cerebral spinal fluid. Okay, so the cerebrum, right? So now we're going to go through the cerebrum and we're going to talk about the lobes and the functions associated with those lobes. So the cerebrum, the cerebral cortex, as we call it, is functionally speaking, this is the center of what we classify as intelligence, and personality and critical thinking. All of those functions, all of the functions that you think about, well, first of all of the functions that are under conscious control so that you are aware of, all of those things take place in the cerebrum. Again, what you think of that makes up you as an individual, all of that is determined by synaptic activity that occurs in the cerebrum. So personality, as I mentioned, critical thinking, memories are stored in the cerebrum, as we'll talk about going forward. So all of those activities happen in the cerebrum. The cerebrum itself is made up of 2 hemispheres that are separated by a landmark called a longitudinal fissure. That's what you're seeing here on this dotted line. And then tracts, again, this kind of circles back to information that we covered last week, but tracts are what we call nerves within the central nervous system. So the hemispheres themselves are also separated, and I should say they're mostly separated, except for there are certain tracts that connect them. So oh my gosh, you scared me. Cereal, I'm doing a lecture. So yes. So sorry guys. So the corpus callosum is made up of tracts that connect the hemispheres, and provides an important way by which those hemispheres communicate. We'll see when we talk about seizure activity that those hemispheres, that when someone has seizure activity that is uncontrolled. If someone just check to make sure you're muted.
So like on some of them, like I got 20, some of them I got like 22, and then like the past like she like inserted them all in,
Hold on, hold
and
on.
I have like 17 on the
Hey, Hannah. Hannah, if you can hear me, could you mute yourself? Thank you. Okay. Well, guys, let's see how many slides. Maybe that's a, well, how are we on time? Let's finish up. Okay, so again, coming back, sorry, that was my dad that walked in who startled me. Let me go back to what I was saying. So the two hemispheres are function pretty much independently in many ways. And the way in which they communicate with each other are through tracks. And the main track that allows them to communicate is what we call the corpus callosum. And again, tracks are nerves. So these corpus callosum, as we'll see when we look at pictures, is white, and that's because of the presence of myelination. Because the corpus callosum, this is where you get the myelinated axons that allow for communication between the two hemispheres. So here are some important features or functions to, I guess, important, not features or functions. How do I want to say this? These are just important things to keep in mind as we're going through and we're learning this material. So we're going to talk about, functionally speaking, how different parts of the brain are associated with specific functions. However, always kind of bear in mind that it is the functions that we assign to specific, for example, lobes of the brain. There's a tremendous amount of crosstalk that occurs, and so it's hard to definitively assign one and only one function to a region of the cerebrum. The boundaries that we find that separate, for example, the lobes are relatively indistinct. Many functions require the coordination of several areas within the brain. And so you'll see that as we go along. I'll say, oh, this structure does this. And then we get to a different structure and I say, oh, this structure does this also. And that's because many of the functions are, the coordination between these different areas is required in order for the function to take place. When it comes to innervation, so the hemispheres themselves are innervated, or the control from one hemisphere, or the hemisphere, for example, the left hemisphere controls the right side of the body. The right hemisphere controls the left side of the body. And that's because there is this crossing over that takes place as nerves leave each area of, so you have the cranial nerves that leave the left side of the brain, they cross over and innervate structures on the right side of the body, and vice versa. And we see this manifesting itself clinically a lot when an individual has a stroke, right? So if somebody has a stroke and it's a left-sided stroke, that the clinical manifestations of that occur on the right side of the body. So if it's a stroke that damaged, that damaged the somatic motor neuron activity to the right arm, to the arm, it'll be the right arm that has some degree of paralysis, not the left. Or if it's, the area of the brain that was damaged controls movement of the eye, it'll be the right eye that has that loss of function if the left side of the brain experiences the stroke. And then finally, when it comes to discussing the hemispheres, there is this phenomenon called cerebral lateralization. So the hemispheres are mirror images of one another. However, there are some functional differences associated with them. For example, speech is controlled by the left hemisphere. And so anyway, and we'll go through that in some great detail, but this term lateralization, cerebral lateralization, that is the term that we use to describe the phenomenon by which the hemispheres are not, even though they anatomically are mirror images, or mostly mirror images of one another, there are functional differences associated with them. Okay, so I'll stop here to give people some time. Yeah, I'll stop here. We're a little bit early, but there were a few of you that reached out that had questions, so I want to make sure to give time to answer those questions, and then we'll pick back up and finish the cerebrum and the rest of chapter 13 on Monday.